Our next speaker is Olivier Preynat-Seauve. Olivier Preynat-Seauve is a doctor of pharmacy, academic researcher, and private docent working at the clinical platform of cell therapy, University Hospitals of Geneva, and at the Faculty of Medicine of the University of Geneva in Switzerland. Olivier has over 20 years of experience working at the interface between stem cells, regeneration, and immunity, with a particular interest in tissue engineering. His main current focus is the study of the regulation of adipose-derived stromal cells in various environments, including the healing process. In addition to his academic achievements, his research has led to several patents based on innovative stem cell solutions. Olivier, if you are ready, the floor is yours. You can share your screen.
Thank you very much for the invitation to present this project. It is a very technical presentation based more on methods rather than science. And the goal is to show a new method we developed in the lab called the AirLiwell technology, which provide a unique opportunity to produce organoids in air-liquid interface conditions. It was initially developed at the University of Geneva and also the Geneva University Hospital, and we are currently developing the application for large-scale manufacturing of this technology. As you know, and very quickly, organoid-based models are widely used, and especially in oncology research. You can have several samples to create organoids. It can be biopsies, tumor biopsies for sure. It is possible to derive tumor cell lines derived from these biopsies and to create also spheroids, or why not organoids based on the use of these cell lines. Genetically modified cells can be used also to create these organoids. Of course, pluripotent stem cells can be used to mimic some tissue around the tumor, for example. Anyway, many mixtures of different cells, including immune cells, vessel cells, etc., can be co-cultured to create organoids, creating a very large amount of descriptions in the literature with many organs, many tumors today are modelized in vivo in organoids models. More specifically applied to the research in oncology, a big challenge today is to mimic the tumor microenvironment. A tumor is not only composed of tumor cells. A tumor is a very complex tissue, including also vessels, immune cells, etc. And a big challenge to really mimic the tumor and to allow the development of personalized medicines is to reproduce this complexity in vivo in our organoids. Today, several options try to recreate this real tumor microenvironment in vitro. Different technical options. Tumor fragments can be derived from a tumor, or tumor cells, or immune cells, more or less separated in different components, and different techniques try to mix and to create organoids mixing these different cell populations. It is not always easy to create at the same time an organoid directly with the immune cells, directly with the tumor cells. Sometimes it is necessary to make separations and to first on one side derive the immune cells, in other side derive the tumor cells, and try to do the mixture. Different other technical options try to culture directly the fragment in glasses or in air-liquid interface to try to reproduce directly the maintenance of a fragment. The advantage is that the similarities with the biopsy is very high. However, in general, in these situations, the stability of the fragments in vitro is quite low, and it is frequently complicated to maintain them in a long time. Today, 6 main methods are widely used to cluster cells, for example, tumor biopsies into 3D structures. A very old method is called the hanging drop method. It is the principle is quite simple. The idea is to create very small drops of cell suspensions in a plastic and to inverse the plate. So each drop of liquid will be kind of liquid microwells. The cells by gravity will sediment at the base of the drop, and we will create in these very small drops organoids. It's quite simple. Unfortunately, it is very heavy. It's a very time-consuming procedure. Sometimes it can be very useful, but for large-scale studies who needs a high degree of homogenization, this hanging drop method is not an optimal technology to produce the organoids. Many methods try to force cells to float and not to adhere to the cell culture support. Many commercial devices propose non-adhesive plates for cell culture. And if the cell is not able to adhere to the bottom of the plate, some spheroids will create. It is widely used. Ultra-low attachment plates are widely used. One main inconvenience of this method is the heterogeneity of cells. It's not always easy to obtain organoids that are homogeneous in size and shape, but sometimes it can be used. Micro-modeling in micro wells that are not adhesive is a widely used method. The idea is to create wells in scaffolds that not create adherence to cells. So the cells are clusterized at the bottom of these microwells that do not adhere to these microwells, so they create spheroids. Many commercial devices today propose this technical option to create spheroids. Magnetic leviation is an old method. It is a very heavy procedure, very expensive procedure. When by the use of iron microbeads and by the use of magnets, you prevent sedimentation of the cells. The cells are not at the bottom of the plates, but in suspension in the plate, and you create organoids. Today, magnetic leviation is not really used in practice in many labs. Also, another method is the rotation of cell cultures. You are creating in a volume cells that are under agitation. During this agitation, the cells cannot, by gravity, go to the bottom of the plate, and spheroids are forming. Again, in this methodology, the level of heterogeneity of the cells in size and shape is very high and can create limitations for the biological interpretation of the data. One advantage of this method is the possibility of scaling up with very large cell volumes. Finally, some extracellular matrix-based methods are available. Some membranes, natural scaffold, are used to mimic the basal membranes. And if you put your cells of interest in these scaffolds, they have the tendency to cluster into 3D structures. So in summary, many methods, depending of the project, of the researchers, of the needs, but each method can have its own limitations and its own advantages. Classical limitation is the adhesion of spheroids to cell culture supports. Also, many supports are commercially available and are ultra-low attachment plates. Sometimes the attachments still exist at a basal level, and sometimes we can observe a small background of attachment of the spheroids in the supports. In this slide, you have an example, a past example in our lab. We were trying to generate spheroids in this situation from mesenchymal stromal cells, and even we used ultra low attachment plates. If many spheroids were not attaching to the plates, after a certain delay of culture, some spheroids created low attachments, and it was a strong limitation for the experiment. So in this situation, typically there is a need to use supports that do not have adhesion capacities with the spheroids, and typically the agarose is a very well known and very practical scaffold to prevent disattachments of the spheroids. Sometimes the limitation is not the attachment, but it is rather the heterogeneity. You have there an example from our lab in the past of a neural organoid culture in suspension. In this situation, we use ultra-low attachment plates under agitation. In this situation, we are not exposed really to attachment of the organoids to the plate, but clearly, after long-term cultures, you have this typical picture of beautiful organoids, but some organoids are very large and some organoids are very small. It's very typical, and more you are waiting your culture, more time is ongoing, more heterogeneity is increasing. Very importantly, the biology, the transcriptomes, the secretomes, the cell regulation in large size organoids is absolutely not the same than small size organoids. So in your data interpretation, you have an average of organoids, but this biological heterogeneity is very strong and impair the interpretation of your data. Another example of technical limitations, organoids fusions. In this picture, again, in our lab in the past, we were studying spheroids from mesenchymal stromal cells, and independently of attachment to the plate, sometimes organoids have the tendency to fuse and to create these very big dark aggregates, plenty of dead cells, and of course, totally impairing the quality of the data. A very common limitation, it is more a biological limitation. If you generate in your culture large size organoids, it's very frequent, especially with cells that have the tendency to grow. You very frequently create necrotic cores at the center of your large size organoids, because oxygen diffusion at the center of the organoid is very complicated when the size of the organoid is large. And of course, at the end, you will study a lot of dead cells in your assay. So prevention of necrotic cores is a very important issue to create organoids. So in summary, today we don't have any ideal method to produce organoids in vitro, but an ideal method would allow very homogeneous organoids, which is absolutely crucial for the robustness of data. Organoids that are stable, physically stable, to prevent their fusion. So I need individualized, stable organoids and not organoids that have the tendency to fuse in my culture. Of course, no adhesion of organoids to the supports used in the cell culture to prevent attachments and changes in their biology and derivation of 2D cultures in my supports. Ideally, I would prefer not to introduce agitations in my organoid culture, because we know that agitation creates a kind of physical stress in the culture, changing potentially the biology of the cells. If the change of medium is easier, it's really important for large-scale applications, especially applications that are trying to test compounds or personalized medicine applications. If I would be able to do that in medium or high throughput screening system with a very simple possibility to change the medium, it would be really practical for this kind of applications. So at the University of Geneva, we have developed the AirLiwell technology. So the AirLiwell technology is a unique opportunity to stabilize organoids in micro-molded wells that are not adhesive. It is based on agarose. So the organoids are individualized, are stabilized physically in the system. Importantly, we introduce in this system air-liquid interface conditions. The principle of air-liquid interface is that your organoids mixed to the microwells are floating on the cell culture medium, and a porous membrane, which is made of polytetrafluoroethylene, is at the interface between the organoids and the cell culture medium. So in this situation, after different technical adaptations, the liquid by capillarity is crossing the agarose gel and is covering the organoids and or spheroids by a nanofilm of medium, establishing air-liquid interface conditions and favoring a lot of exchanges with the gas of air. So we have many advantages in this situation. The degree of standardization is very high because each organoid is stable and individualized. So the size and shape of the cells is very homogeneous. The media changes are very simple because you have this kind of insert floating on the liquid. It's very easy to remove the inserts and to replace the medium. The cell culture is very stable because the organoids are individualized and adhesion to the support and cell fusions are prevented. And very importantly, we have in this situation the contribution of the air-liquid interface. First, it is well known that air-liquid interface prevent necrotic cores because gas diffusion and especially oxygen diffusion will be very strongly increased. First, but also air-liquid interface is well known to change the biology of the cells. Many epitheliums, many cells grow in a more physiological manner when establishing air-liquid interface condition, probably due to the exchanges with the gas of air, but not only. So many applications of cell cultures prefer this kind of environmental conditions rather than immersion. So we have created, to do this technology, pads to mold an agarose gel to make the microwells. It was done by an internal collaboration with Stéphane Durual. So 3D printing allowed us to create this kind of pads. The pads were used to mold a gel of agarose that was heated directly in the insert containing the membranes. And we created these molded microwells of agarose directly on this semi-permeable membrane of polytetrafluoroethylene. We were able to design different shapes of pads to create different shapes and size of the microwells. More recently, through a collaboration with the Swiss Center for Electromechanics, we are also able to create pads by 3D printing of metal. It is a very important issue, technical issue, to favor the scaling up, the industrial scaling up of the technology, especially for 96 well plates. So historically, we first tested in the past this technology with adipose-derived stromal cells, which are kind of mesenchymal stem cells. it is very complicated to create spheroids of mesenchymal stem cells because mesenchymal stem cells produce many integrins and many extracellular matrix proteins, creating a very high level of adherence to supports of cell culture, many high levels of fusions, many high levels of attachment to many things. So using this technology, we tested different cell lines and realized that the degree of homogeneity was very high, creating many homogeneous cell cultures of these organoids. You have a higher magnification of this, showing that in this situation, no fusion, a high degree of standardization of and immobilization of the cells. No necrotic course were observed in these spheroids because they were the size was absolutely controlled, and the air-liquid interface conditions provided oxygen diffusion. Historically, we also tested the technology in neural organoids, because in neural organoids derived from pluripotent stem cells, we had this very strong imitation of heterogeneity in size and shape. We compare this technology with the culture of organoid in immersions, and the results were very clear. Compared to conventional methods to derive these organoids in suspension, again showing this high level of heterogeneity, the AirLiwell technology was producing very stable and very homogeneous in size and shapes of neural organoids without necrotic course. Biologically and very quickly, it was interesting for us to compare organoids derived in immersion versus organoids derived in air-liquid interface conditions. Transcriptomes were very different, and performing single-cell RNA-seq to compare organoids in immersion and organoids in air-liquid interface, there was a clear advantage in the purity of the cells. Neurons were really more pure after using air-liquid interface conditions rather than using immersion.
Olivier, your
yes.
time is up. If you can wrap it up.
For sure, yes.
Thank you.
So just in addition to several characterization, we are currently developing today the adaptation of this technology to non-small cell lung carcinoma cancer, with the goal to create, mimicking the air-liquid interface condition of the lung, a personalized medicines for lung carcinoma. So today, and to end this presentation, we are working with my collaborator, Sanael Aran, to the automated and industrial scaling of the technology. We are able to make at the Faculty of Medicine of Geneva, in a semi-automated manner, 6 well plates and 24 well plates. We are currently working on 96 well plates, with the goal to create at the end the commercial availability of this technology. So for more precision, you are invited to contact my collaborator, Sanael Aran, who is developing specifically this industrial scaling of the technology. Thank you very much. Yeah.
Thank you very much. Maybe we can take one question because we're a little behind on schedule.
Yep. Thank you. There's one question is, as you already mentioned, you have a gas from the top and the liquid from the bottom. Do you see a polarity in the organoids in your system? I think it's difficult to assess, but
I can answer to your question indirectly. We know, we did not check that specifically in organoids, but we know when we are creating a neural organoid in air-liquid interface conditions, not in the AirLiwell, but in a more conventional air-liquid interface conditions, there is a clear polarity. The neural progenitor cells have the tendency to be close to the liquid, and the neurites and mature neurons have the tendency to be close to the air components. So there is a clear polarity showing that mature neurons are more attracted to put their neurites towards the air.
Thank you.